Hello, is anyone there? Yeah, actually, 11 of you are there. My apologies for being late. The security system at Clark would not allow me to sign in. I kept trying to sign in and it said, "We have called your phone and you are not answering." And of course, there was no call for me to answer. So I tried calling IT department and couldn't get through. And anyways, it's just been a mess. My apologies once again. Are there any questions before we begin?
No.
All right, thank you. Somebody confirm that I know you're hearing now. Um,
Yeah, you sound fine. Sound good.
I couldn't log in. The security system wouldn't let me. Once you try to log in, they have a new system in place. You can't even access Canvas. You have to verify who you are over the phone. And I couldn't get the phone call.
That's okay. It wasn't that long. We were just,
I'm about 11 minutes late. Let me say that I've done some grading. Let me share my screen here. So take a look at the grading. I got the last lab and the last lecture graded. I have not succeeded in grading any of the labs for the second time. I was hoping to get started on that this weekend. What is today? Today is April 30th, and we're supposed to be on chapter 4, prokaryote and eukaryotic cells. I think we did start that. Let me warn you, you've got the next quiz, quiz 2, which is due by 11:59 p.m. Wednesday night. You can start as early as 11:59 p.m. Tuesday night. It is a timed quiz. It covers chapter 3 and the end of chapter 2, as well as lab 3 and 4.
I have a question.
Uh-huh.
Would you allow to open it earlier tonight instead of 11:59 p.m. tonight?
No, I have to have it for open for only 24 hours. This is a decision made by the biology department to reduce cheating. Why do you want it to be open earlier?
Okay. It's because it's midnight.
Well, can you take it sometime tomorrow?
I'll try my best in the morning because I have class in the afternoon.
Okay. Usually there's a link showing up here allowing me to jump to where we last were, and this doesn't seem to be working either, and the background is wrong. Both Microsoft Word and PowerPoint, the background is wrong, and I don't know what's wrong. I tried straightening it, and actually I got in Word that now this is white and the print is dark, but the background here is still black. So I'm not sure what's going on. Let's see. Not seeing what I want. Oh, there it is. All right, I have that we were talking about the prokaryote cell arrangements, and we discussed earlier in an earlier slide the arrangements that you find in cocci and the arrangements you find in bacilli. Let me state that with Spirilla, there's only one arrangement, and that is they are singlets. Any question about any of that? All right, here's a typical structure of a typical prokaryote cell. Everything in red you can find in all prokaryotes. Actually, all cells have three components you always find in a cell, and that is the plasma membrane. All cells have a plasma membrane. All cells have cytoplasm, and then all cells have DNA. You could also say a fourth thing, all cells have ribosomes, but do realize that eukaryote ribosomes are different than prokaryote ribosomes. The major difference is eukaryote ribosomes are larger. Well, let me blow this slide up. So all cells have cytoplasm. That's the liquid portion of the cell. In a prokaryote, it's all the inside of the cell. Ribosomes are structures that are in the cytoplasm, but they do not dissolve in the cytoplasm. And that's because they are too big to dissolve in the cytoplasm. Plasma membrane, something that all cells have, as well as all prokaryotes. The nucleoid region is where the DNA sits in the prokaryote cell. The nucleoid region is only found in prokaryotes. In the eukaryotes, we have a nucleus, which is a membrane-bound organelle. We do not have that in a prokaryote cell. The DNA is actually floating in the cytoplasm in a prokaryote. And wherever the DNA floats to, that is where the nucleoid region is, meaning there's no boundary between the cytoplasm and the DNA. It's wherever the DNA floats to. Normally, the DNA is dissolved in the cytoplasm, but at least eukaryote cells, I'm not sure about prokaryote cells, but when eukaryote cells go through either mitosis or meiosis, the chromosome condenses, and then it will precipitate out of the cytoplasm. Some other things that some cells have are a pilus, like the sex pilus. We'll talk about that when we talk about genetics. Some cells have a capsule that is outside the cell wall. Some cells, most prokaryotes have a cell wall, but not all prokaryotes have a cell wall. Actually, most eukaryote cells have a cell wall. It's just that animal cells do not have a cell wall. But all plant cells, all fungi, some protists, and then most bacteria and archaea have a cell wall, but not all of them. A plasmid is something you find in many prokaryotes, not all prokaryotes. I'm not even sure if it's most prokaryotes. It might be just some prokaryotes have a plasmid. A plasmid is a extrachromal piece of DNA. It's similar to the DNA of a prokaryote chromosome. It's just that it's much smaller. And it's obviously outside the chromosome. Animal cells do not have plasmids, but yeast cells do. Some cells have a flagella. If you're a prokaryote cell and you can move around, like swim in solution, then you have a flagella. Fimbriae are hair-like structures coming out of the cell. Cells tend to use fimbriae for attaching to things, like some human pathogens have fimbriae, which attach to our cells. I did talk about the capsule. All right. I think that's everything except for the inclusion. The inclusion is a reserve deposit in the cytoplasm that has become so concentrated that the substance, what's the word again? Loses the ability to be in solution, precipitates, that's the word I'm looking for, precipitates out of solution in the cytoplasm. And then you see this inclusion body when you look at the cell, come on right there. And it's, like I said, just a reserve deposit, whatever the cell is accumulating, and it becomes so concentrated that there's so much of it, it can no longer be held in solution, and it precipitates from solution. And that would be an inclusion body right there. Any questions about any of that? All right, let's talk about the glycocalyx. It is something secreted by some prokaryotes, but not all prokaryotes. It is outside the cell wall. It's usually sticky. It can help protect the cell. There are three types of glycocalyxes. It's also called the glycocalyx. Both pronunciations are correct. The capsule is a neatly organized glycocalyx that has a lot of stuff in it, and it helps prevent phagocytosis. So a bacteria cell that has a capsule cannot be phagocytized by a white blood cell, at least not easily. So the capsule can aid in the bacteria's pathogenicity. The slime layer is similar to the capsule, except that it's mostly water, it's unorganized, and it's loosely attached. A slime layer you cannot see under the light microscope. To see the slime layer, you have to use an electron microscope. And I think the reason why you can't see it is because it's mostly water. And so it just doesn't show up with the light microscope. The capsule you can see with the light microscope. So in microbiology, usually when we're talking about a glycocalyx, we're talking about a capsule. There is a third type of a glycocalyx. It's only found in biofilms. It's the extracellular polysaccharide, which is what a cell secretes. And you have lots of these different cells secreting this extracellular polysaccharide, and then that makes the biofilm. Any question about any of that? All right, we really won't talk much about biofilms. Biofilms are harder to treat if the bacteria are inside a biofilm than if they are not inside a biofilm. One example of a bacteria, let me go back, of a bacteria that has a biofilm is the bacteria that make plaque on your teeth. And the plaque is actually part of the biofilm. Any question about any of that? So we really won't talk about a slime layer in this class because you can't see it unless you use the electron microscope. But we will talk about the capsule quite a bit. All right, I've already mentioned that if a cell has a bacteria or archaea cell has a flagella, then this cell can move. It can use the flagella for locomotion. Usually the flagella allows the cell to swim in a fluid media. It could be a semi-fluid media, and we'll talk about that in the lab. The flagella is mostly outside the cell wall, but there is a portion of it which is actually inside the cell wall. a small portion right here. Come on. So this is a flagella there. And there's a portion of it inside the cell wall. There's a small portion of the flagella that's actually inside the plasma membrane. We really won't talk much about the filament or the hook or the basal body, but we will mention that flagella are made of one protein, flagellin. Any question about any of that? There are 4 basic arrangements of the bacterial flagella. You don't really need to know this. I won't quiz you on it, but you do need to know it for the lab, which I think you guys have already completed. And that is if there's a flagella on one end and there's only one, we call it a monotrichous bacterial flagella. If there's 2 flagella on both ends, it's amphitrichous. If there's flagella on both ends, but there's multiple flagella, it's lophotrichous. And then if there's flagella all over the bacteria, we call it peritrichous. As I mentioned in the lab, when the bacteria are first putting out the flagella, initially they'll be having 0 flagella, no flagella, and then they'll put out monotrichous flagella, they'll put out 1, then they'll put out 2, then they'll put multiples out on both sides. And if they're a peritrichous bacteria, meaning they can make it a peritrichous, they'll put it out all over the cell. You classify the flagella arrangement by the highest classification you see for the species. You do not say it for, I don't know, one of them. You say it only for the highest. You don't say it for all of them either. Okay, because you will always find different numbers in bacteria when you're seeing them, because when the cell first goes through division, the cell will have no flagella. And then it'll put out 1, then 2, and then etc., depending on how many flagella the bacteria can produce. So if the bacteria is a prokaryote, if it can move, that means it has a flagella, because that's the only structure bacteria have for locomotion. Taxis is the movement of the cell either toward or away from a stimuli. There are 2 types of taxis, chemotaxis and phototaxis. Chemotaxis is when the cell responds to a chemical signal and then either moves toward the signal or away from the signal. Phototaxis is the same, except that it's occurring to light. And the cell can either move towards light, like a photosynthetic organism will move towards light in phototaxis, but another cell can move away from the light, which actually this picture is an example of that. The light source is over here, and both of these cells are moving this way, away from the light. Any question about any of that? Come on, mouse. For some reason, my mouse isn't working. Let's see if I can go to the next slide. Got that. Replugged in the mouse. Let's see if it works now. You know, I think it's here. It's just maybe I can't see it. Yep. Well, that's not going to be very useful. Let's see if I can get it over here. Let's talk about axial filaments. They're also called endoflagella. This is a special flagella. It's only found in spirochetes. The flagella is anchored at one end of the cell, and the flagella is inside the cell wall. So here, let me see if I can blow that up a little. So here we have the flagella in the light pink. The axial filaments are in the reddish color. The cell wall is around the axial filaments, the endoflagella. And the endoflagella actually wraps around the cell. Shown there. Let me shrink that down now. It allows the cell to move in a special movement. Well, let's see if I can get it. There we go. And it's sort of a corkscrew-like movement. Let me blow that up. Where the cell can move back and forth. Let me show you there. There's a spirochete, and you can see how limber and not rigid it is. It actually folded in on itself and then change moving from north-south to east-west. And what it'll do is just rotate one way to move in one direction, and then it can move in the other way and go back the other way. Any question about any of that? It's called the corkscrew-like movement. So that's the endoflagella, only found in spirochetes. Fimbriae are shorter than flagella. They usually look like mini hairs attached to the surface of the cell. They are not used for swimming. They're mostly used for attaching to surfaces or some object or something. The sex pili is longer than a fimbriae. See if I can blow this up. So here you can see this cell actually has fimbriae right there. Let me blow that up so you can see it better. And it also has a sex pilus, which is longer right there. And the function of the sex pilus is actually to come out of this cell and then skewer another cell and then allow a plasmid to move from this cell to that cell, meaning the sex pilus is actually hollow and it can allow the plasmid to move across from this cell into that cell. And we'll talk about that when we talk about genetics in chapter 8. Any question about that? The plasmid is DNA, and so when I'm saying it allows DNA to transfer from this cell to that cell, it's usually the plasmid that transfers. The prokaryote cell wall is a complex semi-rigid structure responsible for giving the cell its shape. So if a cell is a bacilli, it is because of the cell wall. If the cell is a cocci, it is because of the cell wall. The cell wall protects the cell and prevents osmotic lysis. If you don't know, if you were to put red blood cells into a beaker of water, which some of you may have done in Biology 160, I don't know if they still do that experiment or not, but the red blood cell will take in the distilled water and then the cell membrane will expand. And more water will move into the cell membrane will expand further. More water will move in, and then the cell membrane bursts, killing the red blood cell. The cell wall prevents that from happening, meaning when water moves into a bacteria cell that has a cell wall, the cell membrane will expand up until the size of the cell wall. After the cell membrane reaches the cell wall prevents the cell membrane from expanding further. That will keep more water from moving into the cell because there's no room for the water to move into the cell. So for the water to move in, you have to have room and the cell membrane has to expand. But the cell wall will prevent the cell membrane from expanding any further. And so that will prevent the cell from going under osmotic lysis. And that will protect the bacteria cell from lysis from water. The cell wall is outside the cell membrane, obviously. It contributes to the pathogenicity of some species, like mycobacteria, have a unique cell wall, and part of the mycobacteria cell wall is pathogenic to us. Most gram-negative cells that have a cell wall also have something pathogenic in the cell wall, and it contributes to the pathogenicity of the bacteria. And that doesn't always happen, but it can. The prokaryote cell wall, if it's bacteria, it is mostly made up of peptidoglycan. That is the main molecule in the cell wall of bacteria. Any question about any of that? All right, peptidoglycan is a polymer of repeating disaccharide. The two sugars in the disaccharide are NAG and NAM. Let me blow this up so I can see it. NAG is N-acetylglucosamine, and NAM is N-acetylmuramic acid. NAG and NAM probably mean as much to you as N-acetylglucosamine and the other, and so you may learn either terms. What peptidoglycan is just repeating NAG and NAM in a chain. They're linked together. That's actually a covalent chemical bond linking these NAG and NAMs together. And then the chains are repeated. So there's one on this side, one on that side, and then there's different layers of them. So there will be one layer here, one layer there. The NAG and NAM chains are linked together by short peptidoglycan. Not short peptidoglycan, short peptide chains. So this NAG and NAM chain, come on, mouse, is hooked together to that NAG and NAM chain by a short peptide right there, which actually links that NAM together with, I think, that NAM there. And then the NAG NAM chain here, come on, mouse, is linked together there by this short peptide here, which actually links that peptide together with that peptide. And that's what gives peptidoglycan its name. A glycan is a sugar, and the two sugars are NAG and NAM. And then it has short peptide chains, and that's where the word peptidoglycan comes from. Any question about any of that? Penicillin actually inhibits the peptide lattice linking the cell wall together. And that's how penicillin G, as well as any member of the penicillin family, puts a hole in the cell wall. When the cell is putting down cell wall, it'll make the NAG and NAM and then link the NAG and NAM together with the short peptide chains. The penicillin actually gets in the way. Like penicillin G could get right here, and then this peptide can't get in there and then link these two NAG and NAMs together. That'll actually put a hole in the cell wall. And the cell membrane can protrude out of that hole, and then more water can move into the cell, so that cell membrane will protrude out of that hole in the cell wall even further, and then it'll protrude further until the cell membrane will actually burst from that hole in the cell wall. Any question about any of that? All right, when we look at the cell wall, there are two different types of cell walls in the bacteria. There's the gram-positive cell wall, which is a very thick peptidoglycan layer, shown in purple here. The yellow is the lipid bilayer of the cell membrane. And when we blow that up, there's the lipid bilayer, mostly made up of phospholipid. There are other molecules in it, like obviously the protein. There are other molecules too, like carbohydrates, but mostly phospholipid. Outside of that, we have the peptidoglycan. It's the main molecule in the cell wall. In the gram-positive cell wall, it's a very thick layer upon layer of peptidoglycan. This is only showing 3 layers of peptidoglycan. That's not correct. In the gram-positive cell, there are at least 20 layers of peptidoglycan, and usually more like 100 layers of peptidoglycan. Any question about that? All right, that's the first bacteria cell. We do have some other molecules in the gram-positive cell wall. There's teichoic acid and lipoteichoic acid. Lipoteichoic acid is teichoic acid, which has a lipid portion attached to it. The teichoic acid runs through the peptidoglycan in the cell wall. The lipid portion of the lipoteichoic acid actually has the lipid portion inside the cell membrane. The teichoic acid may help regulate the movement of cations through the cell wall, meaning a cation, that's a positive ion, can actually help move through the cell wall by just moving down the teichoic acid. That isn't needed, by the way, because a cation is soluble in water, and anything that is soluble in water can come right through the gram-positive cell wall. Any question about that? In the gram-positive cell wall, there are polysaccharides in the cell wall, which can provide antigenic variation. I think 0157, E. coli 0157, I forget the other antigen. The 0157 is an antigen that's in the cell wall. Any question about any of that? E. coli 0157 is a pathogenic E. coli. It's not one that you want in your gut. All right, the second bacterial cell wall is the gram-negative bacterial cell wall. It is thinner than the gram-positive cell wall. So there's the cell membrane. Once again, the phospholipid bilayer. Outside of that is the peptidoglycan layer, which is much thinner than in the gram-positive cell wall. But it is made up of peptidoglycan. And then in the gram-negative cell wall, we have a lipid bilayer on the outer portion of the cell wall. It's actually called the outer membrane of the cell wall. It's outside the peptidoglycan. In this picture here, we're seeing the peptidoglycan is only 1 layer thick. That's not correct. In a gram-positive cell, a gram-negative cell, there are usually between 3 layers of peptidoglycan and about 20 layers of peptidoglycan. The point is, it's thinner layer of peptidoglycan in the gram-positive cell, and then you have this outer membrane of the cell wall. The outer membrane is a lipid bilayer, and this lower region of the outer membrane, or the lowest bilayer, the one most inside towards the cell, is the same or very similar to the cell membrane, where the main molecule is phospholipid. However, the outer bilayer of the lipid bilayer of the outer membrane is a little different. It does have phospholipid, and it's probably the main molecule in the outer bilayer, but the outer bilayer has lipid A, another lipid. That lipid A is attached to an O polysaccharide, making lipid A and O polysaccharide actually a lipopolysaccharide. The O saccharide polysaccharide is the hair, the lipid A is the big glob, blue glob. The lipid A is the important part of the outer membrane of a gram-negative cell because lipid A is an endotoxin. Toxins are something which is toxic to us, and that means that all gram-negative bacteria that have a cell wall have an endotoxin, and it's lipid A. Any question about any of that? All right, in the gram-negative bacterial cell wall, we do have other molecules. We do have, obviously, I've already mentioned the phospholipid, because it's the main molecule in the outer membrane. There are also lipoproteins shown here. The lipid portion will be either in the outer membrane or it can attach to the cell membrane. And then the protein portion usually is in the peptidoglycan layer. But the protein can also be in the lipid of either the outer membrane or the cell membrane. And what it'll do is if the protein has amino acids which are non-polar, those amino acids will be on the outside of the protein, and then the polar amino acids will be folded in, so they'll be away from the lipid of the cell membrane or the outer membrane. Any question about that? There's also the porin protein found in the gram-negative bacterial cell wall. It is important because any molecule that cannot get through the lipid bilayer of the outer membrane, and the cell either wants to get it out of the cell or wants to bring the molecule in the cell, it has to come through the porin protein of the outer membrane. The porin protein is a channel protein that allows some molecules to cross, but not all molecules. For example, glucose, fructose, almost any sugar, amino acids, the cell will want those molecules, and they will be allowed through the porin channel protein. Now that molecule has to get through the outer membrane and the cell membrane. The cell membrane is a little different though, and what it does is it has a transport protein for each molecule it wants to bring in or remove from the cell. And that transport protein, at least generally speaking, we'll talk generally in this class, allows only one molecule to move across the cell membrane. So you have a different transport protein to move the sugar, glucose. Then you have a transport protein to move the sugar, fructose. across the cell membrane. But both of those sugars, as far as I know, all sugars and all amino acids can move through the porin protein of the outer membrane. Any question about that? The porin protein can not allow certain molecules to move across. The most important that we're going to talk about would be penicillin G. Penicillin G cannot move through the porin protein of the outer membrane of a gram-negative cell that has obviously a cell wall. And since penicillin G acts on the peptidoglycan layer of the cell wall, penicillin G cannot harm a gram-negative cell. Yeah, we'll leave it that way. I'm wondering if I said that correctly, but I did, so I'm going to leave it that way. I'm not going to explain it, but so if the gram-negative cell has a peptidoglycan layer, meaning it has a cell wall, and you give penicillin G cannot get through the porin protein, and then it cannot harm the peptidoglycan layer. But a gram-positive cell can be treated with penicillin G. Any question about any of that? All right, I think I've talked about this slide. Everything except for the periplasm. The periplasm isn't terribly important. I'm not going to quiz you on it, but it is the region of the cell wall that includes the peptidoglycan. It does not include the outer membrane. I think I've already talked about this one. The gram-negative outer membrane can offer the gram-negative cell protection from complement protein and antibiotics. I already mentioned antibiotics, where penicillin G and some other members of the penicillin family cannot get through the outer membrane of the gram-negative cell. A complement protein. The complement protein cannot bind to the outer membrane of the gram-negative cell and then harm the cell. The complement protein is something that our immune system makes, and we use it to help defend our body from foreign invaders. Complement protein can attach to a gram-positive bacteria and then attack that cell. The complement protein assists the attacking of the cell. Oh, here it is here, 015787. I was trying to remember the other antigen. Is an example of a pathogenic E. coli. You don't want it. And the O polysaccharide is the antigen of the 0157. All right, any question about any of that? So when we look at the gram-positive cell walls and the gram-negative cell walls, the main difference is the gram-positive cell wall has a thick peptidoglycan structure. The gram-negative cells have a thin peptidoglycan layer. The gram-positive cells are more rigid structure. The gram-negative cells are somewhat rigid, but they are not as rigid as the gram-positive cells. Teichoic acid is found in the gram-positive cells. I don't know if I mentioned it. The teichoic acid is mainly just helping hold the layers of peptidoglycan together. That's the main function of teichoic acid. There's no teichoic acid in the gram-negative cell wall. There's no outer membrane in the gram-positive cell wall, and there is an outer membrane in the gram-negative cell wall, assuming the cell has this cell wall. And what's important about that is there's lipid A in that outer membrane, and lipid A is an endotoxin. Any question about the differences? All right, let's talk a little bit about the Gram stain mechanism. The cell wall does allow for the Gram staining characteristics. Mainly the thicker gram-positive cell wall retains the crystal violet iodine complex of the Gram stain, and the gram-negative cell wall is thinner, and the thinner peptidoglycan layer cannot retain the crystal violet iodine complex. In the Gram stain, we first stain the cells with crystal violet. We then apply the iodine. The iodine complexes with the crystal violet, making a larger molecule. And it's that larger molecule, the crystal violet iodine complex, that does not leave the thick peptidoglycan of the gram-positive cell. In the gram-negative cell, the crystal violet iodine washes out of the thinner peptidoglycan in the decolorizing step, and that step has alcohol in it. The alcohol will dissolve the outer membrane of the gram-negative cell. That outer membrane is lipid, mostly lipid, and the alcohol will dissolve the outer membrane. And then we stain the cells with safranin, and that's why the gram-negative cells stain pink from the safranin. Any question about the Gram stain? All right, this slide is showing you the differences between, or the summary of the gram-positive cell and the gram-negative cell. You only need to know the top portion of this table. You can ignore everything with the red, what are those called, hyphens? Gram reaction, the gram-positive cell retains the crystal violet iodine complex. The gram-negative does not. The peptidoglycan layer is thick on the gram-positive cells. It is thin on the gram-negative cell. This is a mistake in this picture. It says it's a single layer. It's not a single layer. It's a thinner layer. Like I said, gram-negative cells typically have between 3 and 20 layers of peptidoglycan. Teichoic acid is present in gram-positive cells, absent in gram-negative cells. Periplasmic space, we won't even talk about that. There's the outer membrane, it's absent in gram-positive cells, is present in gram-negative cells. And then the lipopolysaccharide content, there's none in the gram-positive cells, and there's a high portion of it in the gram-negative cells because it's part of the lipid A of the gram-negative cell wall. Any question about any of that? All right, with that stated, there are some prokaryotes with atypical cell walls. The first one I want to mention is the archaea, which is not a bacteria. If you noticed, I've been talking about the bacterial cell wall. I've not been talking about the prokaryote cell wall because the archaea have different cell walls. Their cell walls are not made up of peptidoglycan. They have pseudomurine, another disaccharide, which is a little similar to peptidoglycan, but it's a different molecule. And some archaea, of course, don't have cell walls. We haven't talked much about it, but some bacteria also do not have cell walls. So the archaea are different, and that's not surprising because they are in a different domain. Any question about any of that? A different domain from the domain bacteria. But there are two atypical families in bacteria which have atypical cell walls. We've already mentioned one, the mycoplasmas, excuse me, not the mycoplasmas, the mycobacterium. We've already mentioned that one. And that is they have a lot of mycolic acid in their cell wall. And the mycolic acid, besides being a thick layer in the cell wall, is makes that cell wall very different. Mycolic acid is a lipid, and that makes the cell wall practically impervious to something, water, and anything dissolved in water. And that means an antibiotic, which dissolves in water, which are most of them, like penicillin, members of the penicillin, and the sulfa drugs, and most of the antibiotics dissolve in water. They cannot harm the mycobacterium. You have to have a lipid soluble antibiotic to harm the mycobacterium. Because of the mycolic acid in the cell wall, we say the mycobacterium are acid fast staining cells. As the acid fast stain will get in the mycolic acid and then will not be removed in the decolorizing step. So the family Mycobacterium differ in their cell walls of the bacteria. Another family that differs in their cell wall are the family Mycoplasmas. The mycoplasmas lack a cell wall. They only have a plasma membrane, similar to animal cells. And like animal cells, which add cholesterol in their plasma membrane to give it strength, the mycoplasmas add a sterol in the plasma membrane to give it strength. That sterol is not cholesterol. Okay? So the mycoplasmas are different, mainly because they don't have a cell wall. If they don't have a cell wall and you stain them with the gram stain, will they be gram positive or gram negative or something in between? Anyone guess? You should be able to know this. The crystal violet iodine complex, what's it get stuck in? I'm not going to tell you this answer. You should know that one.
Isn't it the peptidoglycan?
Yes, crystal violet iodine gets stuck in the peptidoglycan. The mycoplasmas don't have peptidoglycan. So is the crystal violet iodine going to get stuck in it? No, because it's not there to get stuck in. That means the mycoplasmas will stain gram negatively, or as a gram negative cell. And that's true of all cells that lack a cell wall, by the way. All right, you can damage the prokaryote cell walls either by the enzyme lysozyme or by penicillin. We've already talked about penicillin, and it inhibits the peptide links linking the peptidoglycan together. And so if you have peptidoglycan around and the peptide doesn't form a link in the cell wall, you have a hole in the cell wall. And then the cell membrane can protrude out of that hole. Warm water can move in, and the protrusion will just get bigger until the cell membrane pops. Sort of like blowing a balloon inside a box. The balloon, when the box has a hole in it, the balloon will start protruding out of the hole. You keep blowing the balloon, it'll eventually pop from the hole in the box. However, if you were to blow up a balloon that's totally inside a box, the box will prevent you from adding any more air to the balloon. And so the balloon will no longer expand and it will not pop. And that's much the way cells work with the cell wall. You can also damage the cell wall with the enzyme lysozyme. It actually digests the peptidoglycan, the disaccharide in the peptidoglycan. And so with the enzyme lysozyme, you can digest away the cell wall. You don't need to know the terms for either a gram-negative cell or gram-positive cell, but you can get a prokaryote cell, treat it with the enzyme, actually a bacteria cell, treat it with the enzyme lysozyme, and then dissolve the cell wall. And now that cell will be susceptible to osmotic lysis. Any question about any of that? Oh, look at that. My mouse is now working. I wonder why it wasn't working before. I can see it. All right, gram-negative bacteria are not susceptible to as gram-positives are to penicillin G because of the protein, perforin protein, preventing the penicillin G from getting through the outer membrane. That doesn't mean that the gram-negative cell is not susceptible to other antibiotics. For example, streptomycin can get right through the lipid bilayer of the outer membrane of the gram-negative cell. And then, if you don't know, ampicillin is one of the early members of the penicillin family that was discovered. And ampicillin can get through the protein of the channel protein of the gram-negative outer membrane. And then ampicillin can damage both a gram-positive cell and a gram-negative cell. And there are many other derivatives of penicillin which work on both gram-negatives and gram-positive cells. Carboxycillin is one. I've forgotten off the top of my head any others. I just don't remember the name of them. I think there's oxacillin, but I don't think that'll work on a gram-negative cell. I have to look that up, but I don't think it does. All right, any question about gram-negative cells? So don't tell me that a gram-negative cell is more resistant to an antibiotic. It's more resistant to penicillin G and a few other members like penicillin V of the family of penicillin. But there are other families of antibiotics which will work on the gram-negative cell, such as streptomycin. And then there's even members, I'd say at least half the members of the penicillin family, maybe closer to 2/3 the member of the penicillin family, which can act on gram-negative and gram-positive bacteria. It's only penicillin G and penicillin V and a few other members of the family that do not work on gram-negative cells. All right, any question about the cell wall? If not, let's talk about the plasma membrane. It's mainly a phospholipid bilayer. The phospholipids arrange themselves in this type of an organization where the heads are facing inside the cell because the cytoplasm is mostly water. The heads are, that's the term I'm looking for, hydrophilic. They like being around water. The tails are hydrophobic, and so they'll come together. So the lipid portions of the phospholipid will be together. And then the outer layer of the cell membrane, the tails will be facing inside and the heads will be facing outside because what's outside the cell is mostly water. If you don't know, the body or most organisms are 70% water. And so what's mostly outside the cell is water. And then the fatty acid tails are inside, making the lipid bilayer of the cell membrane. There are other molecules in the plasma membrane, including carbohydrates and proteins. We're going to talk about the proteins. There's 3 different types of proteins in the plasma membrane. Peripheral proteins, and they are a protein that are mainly on one side or the other of the plasma membrane. And that's why they're called peripheral. They're peripheral to the plasma membrane. There are integral proteins and transmembrane proteins, and both of these proteins span the membrane. Both a channel protein and a protein transport protein span the membrane. And that's why the channel protein or the transport protein can allow a molecule from outside the cell to move inside the cell. Any question about any of that? A channel protein differs from a transport protein in that a channel protein just opens and then allows the molecules to move through. A transport protein literally pumps a molecule from one side of the cell membrane to the other side of the cell membrane. And we'll talk a little bit more about transport proteins later in the term. Let's continue talking about the plasma membrane. It is a fluid, and we use the fluid mosaic model to describe the plasma membrane. The plasma membrane is a mosaic of phospholipids and proteins. It can have carbohydrates in it as well. And the membrane is as fluid as olive oil. If you don't know, olive oil at room temperature is a liquid, but if you put it in the refrigerator, it will become solid. And obviously, if you're cooking with olive oil, it will become a gaseous. And it's the gaseous olive oil that we smell when we're cooking something in oil, unless you're smelling the food directly. You can't smell the olive oil when you cook with it. In the plasma membrane, the proteins are largely able to move about in the plasma membrane, and the phospholipids are too. For those of you who have taken A and P, you know that when a ligand or a molecule that binds specifically to a receptor in the cell, usually before the signal is brought into the cell, multiple ligands will bind to multiple receptors, and then the receptors will move together in one side of the cell, and then that portion of the cell membrane will be brought into the cell to begin the signal from outside the cell to inside the cell, meaning the signal will be transferred to the genome, the DNA. And then we might have a change in gene expression. And that we'll talk about later, how cells signal each other. But for here, we're just talking about the main portion is that the proteins can move about in the membrane. In fact, every protein that's in the membrane of a cell can move about unless the protein is anchored to either something outside the cell or something inside the cell. If the protein is anchored, then that protein does not move around in the cell membrane. It will remain fixed in position. So a transport protein usually can move about in the cell membrane. But then a protein that is binding the cell to the outer cellular matrix, like the bone portion attaching to the cell which makes the bone, I used to know that term. Osteoblast, maybe? Somebody from A&P can correct me if I got that wrong. You don't need to know the term osteoblast, but you do need to know that the protein may not move in the cell membrane if it's bound to something like the extracellular matrix of the cell, such as the bone matrix and a bone cell attached to the bone matrix. And then usually if the cell protein doesn't move about in the cell membrane, it's usually bound to the intracellular matrix of the cell. The intracellular matrix is a scaffolding of proteins bound together to make the backbone of the cell, particularly a eukaryote cell. But we do have the intracellular matrix in the prokaryote cell as well. Phospholipids are always free to either move laterally from one area to another or to rotate in position. And that's true of a protein that is not bound by either something in the outer cellular matrix or something inside the cell, the inner I just said it. Now why can't I remember the name? Intracellular matrix. Any question about any of that? All right. The plasma membrane shows selective permeability. That means it allows certain molecules to cross, but does not allow other molecules to cross. No large molecules can move across the plasma membrane unless the molecule has a transport protein to move the molecule from one side of the membrane to another. Ions either do not move across the cell membrane, or if they do move, they move very, very slowly. Lipid molecules can move across the cell membrane. However, if you think about it, like a cell and blood, the blood is mostly water. So outside the cell is going to be mostly water. You're not going to find too many lipids outside the cell in blood. And then small molecules can readily move across the lipid bilayer. For us, the two main molecules which can diffuse across the cell membrane are oxygen and carbon dioxide. And as you know, oxygen comes into our cells and then carbon dioxide leaves our cells. Another small molecule which can slowly move across the cell membrane is water. How water moves in to the cell membrane, let me demonstrate that. They blow this up. Water trying to move into the cell waits for the phospholipid here to move over there, and then the water moves in. It only moves in halfway because it's blocked by this layer of phospholipid, but it just waits its time. And what it does, it's in there now, so it's stuck in there, and it's not replaced by a phospholipid. And then it just waits for this phospholipid to move here to there, and then the water can move into the cell. Oops, didn't mean to do that. And that's how water can slowly move into the cell through the cell membrane. That would not be good enough for a plant cell trying to absorb water in its roots. And so what plants do is they have aquaporins, which are transport proteins for moving the water molecule. And that is the main way that water moves into the roots of plant cells. Any question about any of this? If not, I'm going to end it here and I'll see you at 6:30 for the lab. I'm assuming there's a lab. If there isn't, let me take a look here. Yeah, we have a lab today. Oops, it's the wrong day. We still have a lab today. So I'll see you at 6:30 for the lab.